Hello everyone. In this class, we'll study about electrocardiogram, that is ECG. The electrical activities of myocardium is graphically recorded using surface electrodes. The biphasic recording thus obtained is called as electrocardiogram. It is possible because body acts as a volume conductor. Procedure is called as electrocardiography and the equipment is called as electrocardiograph, which is invented by William Enthone. The ECG provides useful information about heart rate, cardiac rhythm, cardiac hypertrophy, conduction abnormalities, ischemia and infarction, and electrolyte changes. Volume conductor. The electrical activities of heart can be recorded from electrodes placed on various specific sites distant from the heart. Electrolytes present in the body fluids aids the conduction of electrical activity, so body acts as a volume conductor. Concept of dipole and vector. The wave of depolarization travels in a particular direction. The outer surface of depolarized part is negative while the outer surface of uh, part yet to be depolarized is positive, thus a dipole is created. The dipole has a particular direction, that is negative or positive, and a particular magnitude. The magnitude and direction of dipole can be expressed by a vector known as dipole vector. Heart is a functional syncytium. Many small dipoles form in a particular direction. The sum of all of them is called as a resultant of dipole. The resultant of dipole changes in direction and magnitude from moment to moment. The summated resultant vectors over a period of time are taken into account and these are called average vector. ECG is analyzed on the basis of number of average vectors during passage of cardiac impulses. As the wave of depolarization spreads along the heart, the following average vectors appears sequentially. The vector of atrial depolarization is directed downwards and to the left, whereas ventricle has four main vectors. First ventricular vector is because of the ventricular depolarization and it's directed obliquely downwards from left to right, as the left wall of interventricular septum is depolarized first and the wave of depolarization passes across the septum from left to right. The second is ventricular vector is because of the passage of cardiac impulse towards the apex and it is directed downwards. The third ventricular vector it is because of the depolarization of the left ventricle and it is directed downwards and left because the wave of depolarization proceeds from subendocardium to epicardium. It is the largest because left ventricle has the largest muscle mass. The fourth ventricular vector it is because of the depolarization of the bases. It is directed upwards towards the base and small in size. The resultant of all four vectors is called as mean electrical axis of heart. Normally it is directed downwards and to the left and makes an angle of about 59 degree with a horizontal. The angle may vary between -30 to 110. Ventricular depolarization takes place from epicardium to endocardium, so T wave caused by ventricular repolarization is upright. This diagram shows the depolarization of the ventricles. So initially the septum, then the apex, then it is going to spread to the ventricles, and in the end it is going to go to the base where the vector is small. ECG paper. ECG is recorded on millimeter square graph paper moving at a speed of 25 millimeters per second. It is divided into 1 millimeter square by thin lines. Every fifth line is thick both horizontally and vertically. Vertical lines are time calibrations, that is 1 millimeter is equal to 0.04 seconds on x-axis. So time duration between two thick lines is 0.2 second, and vertical amplitude of wave is measured in millimeters. So a deflection of 10 millimeter represents 1 millivolt. ECG leads. There are 12 leads in ECG. We have bipolar limb leads, which are also called as standard limb leads, that is lead 1, lead 2, and lead 3. Then we have augmented unipolar limb leads, which are denoted as AVR, AVL, and AVF. And you have unipolar chest leads V1 to V6. Bipolar limb leads. When both the electrodes of lead are active and potential difference between them is recorded, the lead is then called bipolar lead. Each bipolar lead has one positive and one negative electrode. When the vector is directed towards positive electrode, uh, the upward deflection is obtained and vice versa. Entho1 selected 3 points in the body, that is left shoulder, right shoulder, and mid-inguinal point. When the above points are joined together in an equilateral triangle, an equilateral triangle is obtained. Heart is assumed to be in its center, and this triangle is called as Entho1's triangle. The position of positive and negative electrodes of the bipolar leads are shown in the figure. This diagram showing the different bipolar leads between the left arm, right arm, and the left foot. The lead 1 is between left arm and right arm, where the positive is on the left arm and the negative on the right arm, whereas lead 2 is between uh, the left leg and the right arm, where positive terminal is situated at the left leg and the negative at the left arm. The third limb lead is between the left arm and left leg, where the positive arm situated on the uh, left leg and the negative on the left arm. If the electrical potential at left arm, right arm, and left leg are designated as VL, VR, VF respectively, the voltage recorded in the lead 1 will be VL minus VR, and lead 2 will be VF minus VR, and lead 3 will be VF minus VL. And if you combine together, the voltage, the summated voltage will be equal to 0. It would have been 0 if the right arm is positive and the left foot is negative. Entho1 deliberately changed the polarity of lead 2 in order to get an upward deflection in all the 3 leads. So then the lead 2, uh, he he just changed the polarity, and he made it VR minus VF. Now the voltage across the nth one triangles becomes 0, where the lead 1 plus lead 3 minus lead 2 is equal to 0. That means lead 1, lead 2 is equal to lead 1 plus lead 3. At any given instant, the sum of potential in the lead 1 and lead 3 equals to potential in the lead 2. This is called as Entho1's law. So the maximum amplitude is obtained in L2. Unipolar leads. Unlike bipolar leads, unipolar leads has only one active electrode, and it is called as exploring electrode. The other electrode is kept at zero potential, and it is called as indifferent electrode. So unipolar leads records absolute electrical potential under the exploring electrode. Depending on whether the lead is placed on the chest or the limb, the unipolar leads are classified as unipolar limb leads and unipolar chest leads. Unipolar limb leads. An electrode is placed on the distal part of any limb, that is right arm, left foot, and the other electrode should be at the zero potential. The indifferent electrode is designated as V. So the leads are designated as VL, VR, VF. Indifferent electrode is made by connecting three limbs, that is right arm and left foot, through a high resistance of 5000 ohm uh, to a common terminal which is called as Wilson's terminal. These unipolar limb leads gives waves of very small amplitude. So these leads were augmented by removing the contribution of respective limb uh, to the central terminal. This arrangement has increased amplitudes of waves. So it is almost the 3/3 of the VR, the augmented limb leads, and these are called as augmented limb leads. The augmented limb leads are AVR, AVL, and AVF. So in this diagram, it shows the unipolar leads and augmented limb leads, how they are designated. In unipolar leads, all the three leads are connected and the potential difference is seen between the indifferent electrodes, that is on the okay. Whereas in augmented limb leads, uh, the uh, the two limb leads are connected to the high voltage and the potential difference is sought between this indifferent electrode and the exploring electrode. Unipolar chest leads. These are the names as V1, V2, V3, V4, V5 and V6. The position of exploring electrodes of the various chest leads are as follows. That is V1 is in the right fourth intercostal space near the sternal border, V2 in the left fourth intercostal space near the sternal border, V3 in the midpoint between V2 and V4, V4 in the left fifth intercostal space on the midclavicular line, V5 left fifth intercostal space on the anterior axillary line, and V6 on left fifth intercostal space on the mid axillary line. Wilson's central terminal. According to Kirchhoff's law, the voltage at arm, the foot, and uh, the left arm is equal to 0. But it is not exactly 0 as the triangle is not exactly equilateral and heart is not exactly at the center. To tide over this problem, 3 resistance, that is 5000 ohm, is connected to electrode placed at 3 apical points, and they were connected to a common terminal called as Wilson's central terminal. It functions as a zero potential, and the indifferent electrodes are used in unipolar leads. So this is the normal ECG tracing uh, with the electrical activity of the various part of the heart and during each cardiac cycle, and they are represented by P wave, Q wave, R, S, T, and U. Okay. So this is the P wave, first positive deflection, and QRS complex, where Q is the first negative deflection, and we have a second positive deflection as R, and second negative deflection as S wave. Then we have T wave and U, which are both positive waves. P wave, it is due to the atrial depolarization. It represents spread of impulse from SA node to atrial muscles. The duration is less than 0.1 second and amplitude is less than 0.5 seconds. It represents the functional activity of atrial muscles. Abnormal P waves are seen in atrial hypertrophy, hyperkalemia. Sawtooth pattern of P waves are seen in atrial flutter. PR segment. Following P wave, there is a brief isoelectric segment of 0.04 0.04 second due to the AV nodal delay. QRX complex is due to the ventricular depolarization. Its duration is between 0.08 to 0.1 second. The amplitude is between 1.1 to 1.5 millivolts. It is a complex wave at as it consists of an initial downward deflection, that is Q wave, an upward deflection R wave, and a downward deflection S wave. Q wave is the first wave of ventricular depolarization, which is always negative. It is due to the depolarization of interventricular septum from left to right. R wave is the first positive wave of QRS complex, and it is the main wave of ventricular depolarization, and the tip of R wave is sharp and pointed. S wave is the negative wave that follows R wave and ends at J point, also marks the termination of QRS complex. ST segment, uh, following QRS complex, there is a long isoelectric segment. It represents the duration from end of ventricular depolarization to beginning of the ventricular repolarization, and its duration is 0.32 seconds. In ventricular hypertrophy, QRS complex QRS amplitude increases. In myocardial infarction, ST segment is elevated in leads overlying the area of infarct, and ST segment is depressed in the reciprocal leads. J point junction. J point is a junction between the end of S wave and the beginning of ST segment. T wave. T wave is an upward deflection due to the ventricular repolarization. Duration of T wave is longer as repolarization of the ventricle takes longer time. U wave, rarely we get uh, in a normal ECG. It denotes delayed depolarization of papillary muscles. PR interval. It is the interval between beginning of P wave to the beginning of QRS complex. So this includes P wave and PR segment. The normal duration is 0.12 to 0.2 second. It indicates atrial depolarization and AV nodal delay. When Q wave is absent, it may be termed as PR interval, and when Q wave is present, it is called as PQ interval. PR interval is prolonged in heart block and hypokalemia. PR interval is decreased in WPW syndrome and LGL syndrome. QT interval. It is the interval between beginning of QRS complex and the end of T wave. This includes QRS complex, ST segment, and T wave. This denotes the time taken for ventricular depolarization and repolarization. The duration is between 0.35 to 0.43 seconds. ST interval denotes the duration between the end of S wave and end of T wave. So ST interval is equal to ST segment plus T wave. Pattern of ECG in other leads. The pattern is same in L1, L2 and L3, but amplitude is maximum in L2. In AVR, PRT waves are found to be inverted. This is because depolarization curve is moving away from the exploring electrode. In V1, the small r deep west way are seen, but as electrode position is changed from V1 to V5, the height of R increases, but the depth of S decreases from V1 to V6. In V5 and V6, R wave is more prominent. So this is the 12 lead ECG showing lead 1, lead 2, lead 3, the bipolar limb leads, then augmented limb leads like AVR, AVL and AVF. Then we have V1 to V6, which are chest leads. Examination of ECG. You can calculate heart rate. Heart rate is equal to 1500 divided by number of small squares between two R waves, or 300 divided by number of big squares between two R waves. The ECG can also detect the rhythm. A rhythm can be assessed by ECG in EEG by comparing the length of RR interval. So that is all about the ECG. Thank you.